Okay, let's unpack this. Since the 1950s, humanity has produced just an incredible amount of plastic. It reached, what, 360 million tons in 2019 alone?
Astonishing numbers.
And all that stuff, it doesn't just disappear, does it?
No, absolutely not. The World Bank, they estimated about 1.4 billion tons of plastic waste back in 2017. It's just piling up.
Right. And we think about recycling, but the reality, well, it's not great. Only about 8%, maybe 8.4% recycled in 2018.
It's a tiny fraction, a huge problem we're still, trying to get our heads around.
Definitely. And usually we hear about the big stuff, landfills or maybe microplastics in the ocean.
Yes, the visible things.
But today we're going smaller, much smaller.
We are indeed. We're diving into nanoplastics, specifically in bottled drinking water.
Nanoplastics, okay.
And what's really interesting, or maybe concerning, is that these particles are way smaller than microplastics. So small they're incredibly hard to even find and analyze.
Ah, okay. So less understood, but maybe a bigger risk because of that.
That's the potential worry. Yeah, it's a significant, less understood concern.
Right. It's like trying to map tiny currents deep in the ocean.
That's a good analogy.
And to help us sort of navigate this, we're looking at some specific research today.
We are. It's a paper, Characteristics of Nanoplastics in Bottled Drinking Water. Came out in the Journal of Hazardous Materials in 2022.
Okay, Journal of Hazardous Materials. And they looked at actual bottled water brands.
Exactly. 2 different commercial brands. That's what makes it quite relevant, I think.
So our mission today is to really dig into what this study found. What's in the water? What are these particles like?
Precisely. We want to unpack what the research says about these nanoplastics in something many of you drink every day, bottled water.
And crucially, why they might be a bigger deal than microplastics and what that could, potentially mean for you.
That's the goal.
All right, so let's maybe start with the big picture. Plastic is everywhere, we know that. But why focus so much on bottled water here?
Well, if you look at where plastic goes, packaging is just dominant. Between 2000 and 2015, it was something like 35% of all polymer resins produced.
Wow.
And for food and drinks packaging, one plastic really stands out. PT, polyethylene terephthalate.
PT, yeah, you see that on the bottom of bottles.
Exactly. And it's used so much. 75% of PT goes into food and beverage packaging because it's good at keeping things out. Moisture, gases, bacteria.
Precisely. Which brings us to water bottles.
And we use a lot of those. I saw a figure, 7.7 billion single-use plastic water bottles a year just in the UK.
Just the UK, yeah. Globally, it was 391 billion liters back in 2017. It's hard to even picture that volume.
It really is. And most are PT, and they take, what did you say, like 400 years to decompose?
Around 400 years, yeah. It's a time scale that's just almost geological.
Mind-boggling. So how do these nanoplastics actually get from the bottle into the water?
Okay, so that's the degradation process. The PP plastic isn't perfectly stable forever.
Right.
Things like UV light, sunlight, fluorescent lights, and stores heat, just physical abrasion from handling and transport is knocking around.
Exactly. All that can weaken and break the chemical bonds in the long pate-eat polymer chains. And when those chains break, you get smaller fragments, microplastics, and even smaller nanoplastics.
And these can end up in the water inside the bottle.
That's the finding. Previous studies already showed microplastics are pretty common. One found them in 93% of bottled waters across 11 brands.
93%, wow.
Yeah. But finding the nanoplastics, as we said, that's much trickier because they're so incredibly tiny. Less reported on.
So, okay, microplastics are nearly everywhere. Why the specific focus on nanoplastics?
Yeah.
What makes them potentially more worrying?
That's a really key question, and it comes back to that difficulty in detection and analysis. Standard water treatment, the kind your city uses.
Yeah.
It's not designed to remove particles this small. In fact, some treatments, like using UV light to disinfect.
It sounds like a good thing.
Right, but it might actually break down larger microplastics into nanoplastics.
Oh, wow. So trying to clean the water might inadvertently create these smaller particles.
It's a potential, yes, an unintended consequence. And when you think about how many we might be ingesting, there's an estimate, on average, a person takes in over 5,800 micro or nanoparticles of synthetic stuff each year.
5,800.
And get this, over 88% of that is estimated to come from drinking water.
Okay, that's huge. Drinking water is the main source.
Seems to be, according to that estimate. And while microplastics get more headlines, the worry is that nanoplastics, because they are so small, they might pose more significant health risks.
Right. Okay, this is where it gets really critical for the listener. Why? Why would being smaller potentially be more harmful?
Okay, so there are several factors at play. First, just basic cytotoxicity, how toxic they are to cells.
Right.
That's linked to their properties.
Huh.
Size, obviously, but also shape, solubility, surface charge, how reactive they are chemically, even their sort of electronic structure. And because nanoplastics are so tiny, they have a much, much larger surface area relative to their volume compared to microplastics.
Like comparing a pile of sand to a single rock of the same weight.
Yeah.
The sand has way more surface.
Exactly like that. A perfect analogy. More surface area means more places for interaction.
Okay, makes sense.
Which leads to the second point, something called a corona.
A corona.
Yeah, not that one. When these nanoplastics get into your body, fluids, blood, lymph, whatever, they can quickly attract and bind molecules that are already there.
Yeah.
Proteins, lipids, fats.
So they get coated.
They get coated, forming this biological corona around them, and that coating can change how your body sees and reacts to the particle. It might lead to unexpected toxic effects. It's like the plastic particle puts on a biological disguise.
A disguise.
Yeah.
That's fascinating. So it changes how our system interacts with it.
Potentially, yes.
Yeah.
And then there's the surface chemistry itself. Nanoplastics often have lots of reactive chemical groups on that large surface area. These can directly damage important things, like lipids, which make up cell membranes, or proteins, could disrupt cells, maybe damage blood cells.
Yikes. And you mentioned surface charge earlier.
Right. That matters too. For example, some studies suggest positively charged nanoparticles might end up concentrating more in the lungs.
Like they're drawn there.
Sort of, yeah. And potentially taken up more readily by lung cells. That could increase risks there.
Wow. Okay, so size, surface area, corona effect, surface chemistry, charge. It's complex.
It is. And there's more. Cells can actually swallow these particles whole. It's called endocytosis.
Our own cells eat them.
They can, yes. Once inside, they might end up in lysosomes. Think of those as the cell's recycling and waste disposal centers.
Oh.
But if too many of these foreign particles build up there, they can overwhelm the lysosomes, disrupt their function, maybe even cause the cell damage or breakdown.
So they could gum up the works inside our cells.
That's the concern. And all these interactions, the surface reactivity, the cellular uptake, they can potentially trigger oxidative stress, inflammation.
Inflammation, right. We hear a lot about that.
Exactly. And potentially lead to these particles accumulating in different tissues over time. The original paper actually has a figure illustrating some of these potential pathways.
It's quite a concerning picture being painted here.
Mm.
So how did these researchers actually do this? How did they find and analyze these invisible particles in the bottled water?
It was definitely a challenge. They needed a good amount of starting material, so they got 16 liters of water from each of the two brands, brand A and brand B.
16 liters. Okay, that's a lot of water bottles.
It is. Then they used a technique called tangential flow ultrafiltration, TFU.
TFU.
Think of it like a very, very fine filter, filtering across the surface, not just straight through. They used a filter with a tiny pore size, about 1.2 nanometers. It lets water and very small dissolved things pass through, but traps larger molecules and particles, including the nanoplastics.
Okay, so trapping the targets.
Right. And they kept filtering until those 16 liters were concentrated down to just 200 milliliters of what's called the retentate, the stuff left behind.
So 16 liters down to 200 milliliters. That's what, an 80-fold concentration?
Exactly, 80-fold. Very concentrated. Then they needed to get rid of the natural mineral salts in the water because those could interfere with the analysis later.
Ah, right. Salts could crystallize or something.
Precisely. So they used a process, basically dialysis with deionized water, to wash the salts away. And finally, they carefully evaporated the remaining water to get a solid sample of the concentrated nanoparticles.
Okay, quite a meticulous process to isolate them. Then what? How did they figure out what they actually had? Size, shape, what they were made of?
They used a suite of different analytical tools. For size and shape, they used dynamic light scattering, DLS, which measures how particles scatter light to determine size distribution, and also a special microscope called SEML, Super Resolution Microsphere Amplifying Lens.
SEML. Why not a regular electron microscope?
Good question. Apparently, standard electron microscopes can struggle with imaging these tiny plastic particles. They can have surface charges that cause problems, or they just clump together. SEML was better for visualizing individual particles.
Okay, clever. And for identifying what they were.
For the chemical structure, they used FTIR Fourier transform infrared spectroscopy.
FTIR, okay, like a chemical fingerprint.
Exactly. It measures how the sample absorbs infrared light at different wavelengths, giving a unique spectrum or fingerprint for the molecules present.
And you mentioned breakdown products earlier.
Right. To identify the specific chemical structures, especially potential breakdown products from the PET, they used TDGCMS,
Okay, that's a mouthful. TDGCMS.
thermal desorption gas chromatography-mass spectrometry. Basically, you heat the sample gently, thermal desorption, to release volatile compounds. Then you separate those compounds using gas chromatography, GC. And finally, you identify each separated compound by measuring its mass using mass spectrometry, MS.
So heat, separate, identify by weight.
That's the essence of it, yeah. It lets them pick out specific chemical signatures of the plastic in its degraded form.
Wow, a really sophisticated approach. So the big reveal, what did all this tech tell them? What did they find in brand A and brand B?
Well, first, the microscopy, the SESMAL imaging, showed that most of the organic nanoparticles they saw in both brands were generally transparent and roughly spherical.
Tiny, clear balls floating in the water.
Pretty much. Then the DLS gave more detail on the sizes, and there were some differences between the brands.
Oh, yeah.
In brand A, most of the particles, about 84%, were around 433 nanometers in size. There was a smaller group, about 16%, much smaller, around 66 nanometers, and importantly, no detectable microplastics in brand A.
Okay, so brand A was mostly nano, two main sizes. What about brand B?
Brand B was a bit more varied. About 65% were around 140 nanometers. Another chunk, 31%, were larger, around 605 nanometers. And interestingly, they did find a small amount, nearly 4% of particles in the microplastic range, around 5 micrometers.
Ah, so brand B had a wider size range, including some actual microplastics, while brand A was more purely nano.
That seems to be the case based on their measurements, yes.
Okay, so that's size and shape. What about the chemistry? Did the FTIR confirm they were actually plastic? Degraded plastic?
Yes, the FTIR analysis was pretty crucial here. It showed signatures consistent with degraded PE compounds in both brands.
How could they tell it was degraded?
They compared the spectra they got from the samples to the known fingerprint of intact PT. They saw that some absorption peaks, which are usually strong in peat, were weaker in their samples.
Meaning the chemical structure had changed.
Exactly. Specifically, changes suggesting the formation of carboxyl acid end groups, which happens when the long peat polymer chains break. They also saw other subtle changes, like a peak linked to the ethylene glycol part of peat aging, and maybe increased CH stretching vibrations, possibly due to weathering.
Weathering. Even inside a bottle.
Well, weathering in the chemical sense, degradation potentially caused by light and hydrolysis, yeah. And some other typical P peaks were actually missing entirely, further suggesting the original structure had broken down.
Okay, so strong evidence from FTR that it's broken PET. Did the TDGCMS confirm that? Did it find specific breakdown chemicals?
Yes, this was perhaps the most direct evidence. The TDGCMS found phthalate derivatives, which are common plastic additives.
Okay, plasticizers.
Right. But more significantly, in both brands, they found substantial amounts of a compound called ethyl p-ethoxybenzoate.
Ethyl p-ethoxybenzoate. Never heard of it.
Me neither. But the key thing is, this compound was not in their control sample. They're blank. And its chemical structure looks very similar to a piece you'd get if you broke down the PT molecule itself.
Ah, like a specific fragment of the original P chain.
Exactly. That strong structural similarity and its absence in the blank led the researchers to conclude it was very likely a decomposed component coming directly from the PT bottle.
Wow. That really seems to nail it down. It's not just contamination, it's bits of the bottle itself.
That's what the evidence strongly suggests, yes.
And they thought this breakdown was likely due to light exposure, UV light, during packaging, storage, sitting on the shelf.
That was their hypothesis. They mentioned the water was analyzed about 4 months after bottling. That time frame certainly allows for cumulative exposure to light, UV, or even visible light, which can trigger this kind of peat degradation, known as Norrish degradation.
Okay, that makes sense. Now, this sounds pretty compelling, but science always has limitations, right? Did the researchers acknowledge any in their study?
They did, absolutely. Good science always recognizes its boundaries. Firstly, they stressed that they only looked at two specific commercial water brands.
So we can't necessarily say this applies to all bottled water everywhere.
Correct. The findings are specific to those brands analyzed at that time. More studies are needed for a broader picture.
Okay, what else?
A key limitation was quantification. While they showed strong evidence these nanite-sized peat-late breakdown products were present,
They couldn't say exactly how much was there.
precisely, they couldn't quantify the exact amount. The main reason was the lack of a suitable standard material for calibrating their TD-GCMS instrument for these specific nanofragments.
You need something known to compare against to measure quantity.
Exactly. And creating that standard is really tricky. You'd need a material of known mass, super tiny, below 10 kilodaltons, they said, that behaves exactly like the real degraded nanoplastics during the complex heating and separation process.
And ideally made under the same conditions. Oh, yeah, that sounds incredibly difficult to make.
It really is. Developing those kinds of accurate standards for nanoplastic analysis is a big challenge for the whole field.
So they know it's there,
Yeah.
likely from the bottle, but the exact concentration is still an open question pending better measurement tools.
That sums it up well. But even with that limitation, the implications they draw are pretty significant.
What did they suggest?
They explicitly suggest careful consideration of the use of plastic containers for drinking water. That's quite direct language for scientific paper.
It really is. Careful consideration implies maybe thinking twice about it.
It does. And they also stress the need for more systematic, controlled, and randomized studies to really understand how common these nanoparticles are across different water systems.
More research, always more research needed.
But they frame it with some urgency, mentioning the possibility that this is an existing risk. They're not saying it is definitely harmful at these levels, but the potential is there and needs investigation.
An existing risk
Yeah.
that catches your attention. Did they suggest any actions beyond more research?
Yes. They strongly recommend moving towards systematic quantification, figuring out how to measure how much is there consistently. And importantly, they call for establishing national and international standards to actually limit the amount of nanoplastics allowed in drinking water.
Setting legal limits, basically.
Essentially, yes. They highlight the concern about potential cumulative effects over time, even from low levels, which really underscores why timely action and regulation might be needed.
Okay, so summing this all up, this deep dive shows that bottled water, something we often think of as pure, can contain these tiny, tiny fragments of the plastic bottle itself. Nanoscale fragments.
That's the core finding, yes. The bottle itself seems to be shedding these particles into the water through degradation.
And while we don't know the full health effects yet, the science suggests these nanoparticles might interact with our bodies in complex ways, getting coated, maybe damaging cells, causing inflammation, and they aren't easily removed.
That's the area of active research and concern, definitely. Their small size and large surface area make their biological interactions potentially quite different from larger particles.
It really makes you think, doesn't it?
Yeah.
Given how much bottled water people drink globally, what does this make you, the listener, think about your own choices? And this whole enormous issue of plastic.
It's a really pertinent question. If something seemingly straightforward like bottled water has this hidden nano-level complexity, it's worth reflecting on. Maybe you want to look into ongoing research on nanoplastic toxicity, or perhaps investigate water filtration options, or even explore alternative packaging.
Lots to consider. And it leaves us with that sort of final, maybe unsettling thought. If bottled water has these unseen particles, what else might we be ingesting without realizing it? And what are the long-term consequences, not just for us, but for the environment too?
That's the big unknown, isn't it? It certainly suggests we need a much deeper understanding of how these materials behave at the nanoscale in our environment and within our own bodies. Definitely food for thought.